A quick one. My name is Mosjaro. Very quickly, in this lecture series, particularly, I want to be focused on the biomolecules, and this class will be very of good use to persons that are in the medical school, and of course those persons in university from 200 level, 300 level. It all depends on where you are having this particular block of lecture. So very quickly, I want to be breaking it down very fast. I want to be dealing with the biomolecules first. I'm talking about the carbohydrates, talking about the lipids, and of course talking about the proteins. Now we want to look at how these what forms these biomolecules and the necessary points we have to know about these biomolecules. So very quickly, a very quick one, please do well to click on the subscribe button, because there are going to be a high number of lecture series coming up. But we're going to be breaking it down so that you need to understand the particular point that you're having problem with. So we have to be fast but in depth at the same time. So what biomolecule are we going to be looking at today? First off, we want to look at the in-depth analysis of the carbohydrate. Before then, we'll now begin to check out the metabolism of carbohydrate, talking about the glycolysis and all that. Now remember that the three major class of food where we get our calories, our energy from, are the carbohydrate, the lipids, and the protein. Well, of course, the lipids have higher number of calories when it comes to carbohydrate. But what's the significance? Why is that humans rely so much on carbohydrate? Because carbohydrates are easily what? Oxidized. So quickly, we want to look at the biomolecule carbohydrates and see how they are classified. Then before we begin to look at their metabolism, you don't just rush into things. So don't be a fool. Let's now focus. Let's hit it. Now, talking about the carbohydrates. Now, first off, one major word I will want to use to classify the carbohydrate, that the carbohydrate, because of their usefulness in our day-to-day living, is regarded as the staff of life. The carbohydrate is regarded as a staff of what now? Of life. Now, these carbohydrates can be classified into various areas. So I'm going to be breaking it down. So I've heard persons saying, oh, Mosjaro, I have a problem in carbohydrate. No, fear no more, because I'm going to be bringing out a whole lot of lecture series concerning these biomolecules, their metabolism, and what forms them. So quickly, let's begin to take a breakdown. Of course, I'm doing a lot of cleaning today because the class is going to be well summarized and in-depth. Now, there are two. These carbohydrates can be categorized into two. Now, what are they? We have what we call the sugar carbohydrates, and of course, we have the non-sugar carbohydrates. Now, if we are dealing with the sugar carbohydrates, what are we referring to? What carbohydrates moieties are we talking about in this situation? Now, we are looking at the monosaccharides. Now, in dealing with the monosaccharides, we'll have to list out some common examples of monosaccharides. I'm talking about in this monosaccharide, we're looking at the azoose sugars, so to say. Now, what are the azooses? Azoose sugars are sugars that contain 6 carbon atoms. Now, there are some sugars that contain 5 carbon atoms. They call them the pentose sugar. There are some sugar that contain the 4 carbon atoms. Talking about the tetroses and examples of tetrose. We're talking about the erythrose, the erythrulose. Now, we have those that are the triose. Of course, the simplest sugars are the what? The triose. Now, the simplest sugar, you have to pay attention to that. The simplest sugars are the what? Triose. Now, we're talking about under this triose, example of triose, you have the glyceraldehyde. Glyceraldehyde is an example of a what? Triose. It contains 3 carbon atoms. Now, let's look at the monosaccharides. Examples of monosaccharides. Now, under this place, we have the glucose, we have the mannose, we have the fructose, of course, which of course is a ketose sugar. Now, I like to give a quick breakdown. That's why this lecture series is very important. You don't have any fear, so you have to look at it in depth. So we want to look at the major points. So anything you are going to be saying is going to be well simplified for your understanding. Now, this fructose, of course, as a chemistry student, you know the carbonyl group. This is the carbonyl group, of course. Now, this carbonyl group is found in fructose. So because of that, fructose is regarded as a what now? A ketose what? A ketose sugar. Now, specifically, and I want to drop these points, I'm always filled with points, so I don't have time to waste. Now, Ketoses are usually tested for in the laboratory, though we are not looking at tests in this lecture series. That ketoses are usually tested for using a specific test which is regarded as the Seliwanov test. Now, this Seliwanov test is specifically used to test for ketoses, for ketose sugar, and a good example of a ketose sugar is fructose. Now, examples, still moving on, kicking on. With the monosaccharides, now we have glucose, we have mannose, we have fructose, of course, we have galactose. These are examples of monosaccharides. Now, under sugar carbohydrates, we also have the what? The disaccharides. Now, talking about the disaccharides, we are referring to the sucrose. Sucrose is a disaccharide. We are referring to the maltose. Maltose is a disaccharide. We are referring to the lactose. Lactose is a disaccharide. And of course, the cellubiose. Some persons are not quite familiar with these. And of course, we have the mellibiose. Now, these are common examples of disaccharides. Now, there's a problem about the disaccharides. Not just listing out the disaccharides. You must know the monosaccharide. Because say disaccharide, definitely, of course, you should know it's made of what? Of two monosaccharides. Now, first off, we have to know the monosaccharides that forms the disaccharides and the bond that exists between monosaccharides. So we take it from there now. Now let's begin to trash. Now we also have, remember, under the sugar carbohydrate, please don't get it twisted. Under the sugar carbohydrate, we also have another categorization of carbohydrate, which is regarded as the oligosaccharides. Now you see the breakdown. So that's why this lecture series is so in-depth. That's why you should click on the subscribe button, because when Mojaro drops, you have to grab it like the chicken and the corn. Now look at it now, quickly. Now, these are monosaccharides. These are disaccharides. What then are the oligosaccharides? But please, the oligosaccharides are not categorized under the non-sugar carbohydrates. Now, these oligosaccharides are sugars or carbohydrates that are made of 3 to 11 monosaccharides units. 3 to 11 monosaccharide units. Now, we are going to be taking a breakdown of oligosaccharide, but please, there's something I want us to do first quickly, because it's self-explanatory. Like it's so easy. When it comes to Mojaro, you have to follow it and break it down step by step. Now, we have to be looking at the units of monosaccharides that forms these disaccharides. Then we will take our quantum leap very quickly. So oligosaccharides. So quickly grab your seat. Now let's look at the disaccharides and see the type of bond that exists in the disaccharides. Now quickly, we are talking about the disaccharide. Now talking about the disaccharides. Now quickly, let me say this specifically. Maltose, for instance, I will go with maltose for instance. Maltose is made of 2 glucose units. That means glucose plus glucose. We form what? We form maltose. Now, quickly, please, when monosaccharides comes together, now the joining of 2 monosaccharides, there is a bond that exists between a linkage rather, or a bond if you like, that exists between 2 monosaccharides, and those bonds are regarded as what? As glycosidic bond, or you can say glycosidic what now? Linkages. Now, quickly, there are different types of glycosidic bonds and different types of glycosidic linkage. Now, I want to quickly bring this out for us. There are some, if I want to bring out the structure of glucose. Now, glucose can be represented in molecular forms. Of course, the formula for glucose is C6H12O6. Now, because I am joining 2 glucose together, now this is the molecular representation of glucose. Now, I want us to listen carefully. Don't allow anybody to distract you. Anybody trying to distract you, of course, you know what to do. Now, come. This is glucose. Now, I will say that maltose is made of 2 glucose molecules, and there is a bond that exists between the 2 glucose molecules, which is what? Glycosidic what? Bond. Now, be careful. Now, in the formation of glycosidic bond, a water molecule is what? Removed. Therefore, if I want to write the molecular formula of maltose, it becomes C12 H22, it ought to be H24, but because I'm losing a water molecule, and remember that water is H2O, I'm removing one hydrogen. So if I'm adding C6H12O6, so if I'm combining both of them together, of course C6H will be C12, right? Then we have H12, it will be ought to be H24. Then because there's a water molecule that is removed in the formation of the glycosidic bond, so it's H22, then of course O11. Now, that is not the issue. Those are for kindergarten. Those are kindergarten points. But what I'm trying to bring out here definitely is that in the formation of disaccharides or oligosaccharides, now there are some structures that you must know. How are these bonds formed? Like I said just now, we have alpha 1,4, we have beta 1,4, we have alpha 1,6, we can have beta 1,6. Now, don't get it twisted. Relax. Calm your nerves. How are these bond forms? Now, I'll take a case study. There is what we call Haworth projection, or Haworth projection of what? Of glucose. Now, glucose can be represented by the Haworth projection by the Pyranose ring. Don't be scared, yeah? Now, look at the Pyranose ring. Now, I'm going to show you how the Pyranose ring is being what? Is being numbered. Now, look at the Pyranose ring. Now, this is carbon, 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 carbon. Now, if I want to write a structure of glucose using the Haworth projection, now we have to be very careful. Using the Haworth projection, I have to say there are two structures I can bring out. Now, one is this. One is this. Now we have something like this. OH, then we have what? We have H. Then, of course, we have OH, then we have H here. Then we have OH, then we have H. Then, of course, we have OH, then we have H. Then, of course, we have CH2OH. Now, we have to be very careful. In the numbering of the carbon atom, do you know why I have to know how the carbons are numbered? So you get to understand. Anytime I say alpha 1,4, you know what I'm trying to talk about. Anytime I say beta 1,4, you know what I'm talking about. Now, look at it. This place here is one carbon, carbon number 1. There is carbon number 2. Yes, carbon number 3. Yes, carbon number 4. Yes, carbon number 5. Yes, carbon number 6. So this is how the Pyranose ring of glucose is numbered. Now, this structure I just brought out here now is what? Is beta glucose. How do I get to know? Because of the OH here. This OH here is at the top. So you can easily know it. When the OH is at the top, it's beta. But when the OH is below, I'm having something like this now. I'm having something like this. I'm having H and I'm having OH. This one is now alpha glucose. Making sense? So we have what? So you have to look at the carbon number 1. So this is what we're getting. So if it's here, OHH is beta. But when it is, I'm having something like this now. When I'm having H and OH, then it becomes what? Alpha. So if I'm forming an alpha 1,4 glycosidic linkage, what is that? That means I am going to be adding something like this quickly. Sorry, I have to take this off so I get to understand it properly. Now, I come here and I see a Pyranose ring, for instance. You have to take it easy on my structure, but just understand what I'm trying to pass out. Now, we have OH, for instance, and we have H. Now, of course, we have the OH, then we have H, and of course, we have OH, then we have H, and OH, and we have H, and remember, we have the CH2OH. Now, this is beta, right? Now, if this is beta, if I want to combine with another glucose molecule, so because this is beta, if I want to combine with another one, of course, there will be a removal of water to form another. So this kind of linkage, we now say it is what? Beta 1,4. Because, but if it is below, I will not say it is what? The OH is here, the H is here. I will not say it is what? Alpha 1,4. So when we're talking about glycosidic bond, just know. But one thing I want you to catch here quickly. When I say alpha glucose, you know what I'm talking about. When I say beta glucose, you know what I'm talking about. It's not specifically to glucose alone. Of course, we can have that for galactose and of course the other monosaccharides. But quickly, let's now move on straight up to the maltose. Now we've said that maltose are made of 2 glucose molecules. Yeah. Now because it's made of 2 glucose molecules. Now listen up. It's made of 2 glucose molecules. If it's made of 2 glucose molecules, what type of bond exists between the 2 glucose molecules in maltose? We are having an alpha. Look at it now. Alpha 1,4 glycosidic linkage. Alpha 1,4 glycosidic what? Linkage. That is the type of glycosidic bond that exists between 2 glucose molecules. Now I want to quickly combine or compare another disaccharide that contain the same glucose units so that we can easily understand it very quickly. And what is that? Quickly, we are having cellubiose. Now, cellubiose also contains 2 glucose units. So we have glucose and what now? And glucose. Now we have 2 glucose units. Now, these glucose units, but the difference between this and this other one is that when it comes to cellubiose, the glycosidic linkage is what? Is beta 1,4 glycosidic linkage. So when you are studying, you have to be very, very careful as you analyze this. So we have the alpha 1,4 and we have the word beta 1,4. I'm on fire today, that's why I'm sweating. Pay no attention to my sweating. Let's look at it now very quickly. Now, we have looked at maltose. Why am I comparing the maltose and the cellubiose? Because they have the same monosaccharides units. But please pay attention that the glycosidic linkage are quite different. So this one is alpha 1,4 and this one is beta 1,4. Now there's another comparison I want to bring out again, another disaccharide very quickly. Now what disaccharide is that? We have to look at lactose. Lactose, very good. Of course, everybody here knows that lactose is also regarded as the milk sugar, the sugar found in milk. And now talking about lactose, there are some persons that are lactose intolerant. That means they cannot easily metabolize or break down lactose. Now that is a lecture series for another time, so keep yourself posted, don't miss it. So why are they lactose intolerant? What is lactose intolerance, so to say? Now quickly, now if we have said that very quickly, now we have to look at, pardon me, let me put this up now quickly, we have to look at the units that forms lactose. Quickly, lactose is made of glucose linked to galactose. Glucose and galactose, and of course we have the glycosidic linkage. Now what glycosidic linkage exists between glucose and what? And galactose. That glycosidic linkage is what? Is beta 1,4 glycosidic what? Linkage. Beta 1,4 glycosidic what? Linkage. Now there is another disaccharide that have the same monosaccharide units as lactose, and who is that? That is what? Mellibiose. Now what's the difference between mellibiose and lactose? They have the same units, but the glycosidic linkage differs. So please, when you're preparing for your examination, be very careful of the glycosidic linkages and of course the bonds. Now what's glucose? So we're having glucose, of course, linked with galactose, of course, galactose. Now, and what's the glycosidic linkage? Alpha 1,6 glycosidic what? Linkage. So that is the glycosidic bond that exists in what? Mellibiose. Then, I know you are asking me, say, why did you skip sucrose? Of course, sucrose is a very important sugar. We do consume sucrose day to day in our various meals that we take, that we consume. That's also regarded as a common table sugar. Now, what makes up sucrose? Sucrose is a non-reducing sugar. Now, what is the composition of sucrose? Sucrose is made of glucose and the ketose sugar, fructose. Now, there's a problem in their glycosidic bond. Now, let me bring it out very quickly. Now, we are still dealing with the disaccharides. Now, quickly. Now, what makes up sucrose? Now, sucrose as a disaccharide is made of what? Of glucose linked, of course, there's a glycosidic linkage to what? to fructose, which is a ketose sugar. Now, the main mention of fructose just now is a ketose sugar. Now, as the class goes by, we're going to be saying, I'm going to be dropping it very quickly. We're going to be talking about the storage form of what? Fructose. Now, what is the storage form of fructose? It's inulin. Don't be a fool. I didn't say insulin. Insulin is the hormone, of course, helps in blood sugar regulation. I'm talking about inulin. So just like we have storage form of glucose, repeating units of glucose can form cellulose, we can have starch, we can have glycogen. Likewise, repeating units of fructose, we have what? Inulin. Making sense? Now, we also have repeating storage form of galactose. And what's that? We have what they are called what? Galactans. We are not here yet, but you have to program your mind. Of course, when it comes to Monosaccharides, it's all points, all true. I don't like wasting time. I don't like wasting your precious time. Now, look at it now. So, inulin, storage form of fructose. Galactans, storage form of what? Galactose. Now, these are polysaccharides, obviously, but we are not gotten to that aspect of polysaccharide. But let's do the count, relax your mind, and take a deep breath, and let's go back. Now, talking about this, what's the problem? What type of bond is this? Is what? It's an alpha 1 beta what? 2 glycosidic what? Linkage. It's quite complex. It's alpha 1 beta 2 glycosidic linkage that exists between what? Glucose and fructose in what? In sucrose. Now, we have dealt with the what's it called? We have dealt with the disaccharides. We have looked at monosaccharides very fast. We have looked at monosaccharides. Now, quickly, let's pay attention. Let's quickly look at the oligosaccharides. Remember, I told us just now that the oligosaccharides are made. The oligosaccharides are made of 3 to 11. Now, some persons will say 3 to 10. Yes, it doesn't really matter, but specifically, basically, that the oligosaccharide is made of 3 to 10 monosaccharides. Now, watch it. 3 to 10 monosaccharide. I didn't say 3 to 10 carbon atom. Don't be a fool. Now, look at it now. Come. If we are having oligosaccharides, now, there are some oligosaccharides. Of course, we said 3 to 11 monosaccharides. Now, there are some oligosaccharides that are made of 3 monosaccharides. What do we call them? They are regarded as what? Trisaccharides. Now, there are some that are made of 4 monosaccharides. They are known as what? Tetrasaccharides. There are some that are made of 5. We have the pentasaccharides. Now, there are some that are made of 6. We have the word hexasaccharide. I can go on and on. Now, look at what we're going to be doing now. We want to look at this briefly and take their examples. These are the specific areas that you should be focusing on when it comes to your level examination. Now, strictly, without wasting much of our time. Now, let's look at trisaccharides. An example of trisaccharides. Now, pay attention. Anytime we are looking at this oligosaccharide, it is proper that we bring out the monosaccharide units, and of course, yes, the monosaccharide unit that form the trisaccharide. So it's not just enough for me to say, trisaccharides are made of 3 monosaccharides. No, don't be an idiot. Now, you should be very careful. Now, quickly, these trisaccharides, example is raffinose. Yeah, raffinose is a trisaccharide. Now, what are the monosaccharides that forms trisaccharides? We have galactose, yeah, we have glucose, yeah, and we have what? Fructose. Remember, there are glycosidic bonds that exist between these. Now, this is an example of what? Of trisaccharides. Now, talking about tetrasaccharides. Tetrasaccharides. Now, an example of tetrasaccharide is stachose. Don't make the mistake thinking that stachose have to be related with starch. Starch is a polysaccharide, but stachose is an oligosaccharide. Now look at it now. Stachose is made of what? What's it made of? Stachose is made of 4 monosaccharides. And what are the 4 monosaccharides? We have galactose, glue, fruit. What do I mean? Galactose, glucose, fructose. Right? So it's as if stachose is raffinose with an additional galactose. Yeah? So now look at it now quickly. Then of course we have the pentasaccharide. And this pentasaccharide we have the verbascose. Verbascose is a pentasaccharide. The verbascose. Now a good example of verbascose here. I need to remove this quickly. And pardon me. Example. What makes up the verbascose? The verbascose is made of what? 3 galactose, glucose, and of course fructose. Now, these oligosaccharides can be hydrolyzed to form disaccharides and to form smaller oligosaccharides. What do I mean? This can be hydrolyzed, of course, to form what? To form raffinose. Yeah? Now, look at it now. Quickly. Now, we have looked at, this is verbascose. This verbascose, remember, quickly if I clean that off, verbascose is a pentasaccharide. Now, there is what we call the hexasaccharides. Example of hexasaccharide. Now, example of hexasaccharide is what? Is alpha cyclodextrin. There's a question that if I'm watching this video, I would like to ask Ms. Jaro quickly, but of course we don't have that interaction, but I want to help you to think something out. Is it that in oligosaccharides, all the repeating units must be different? This is a case study of in which all the repeating units are the same. So alpha cyclodextrin is made of what? Is made of 6 glucose units. 6 glucose units. That's what's from what? The alpha cyclodextrin. Now, we have succeeded in looking at the oligosaccharide. But remember, we said we have the sugar carbohydrate and we have the what? The non-sugar carbohydrate. Now, talking about the non-sugar carbohydrate, let's take a breakdown of the non-sugar carbohydrate. This non-sugar carbohydrate, we can have the non-sugar, let me bring it out. The non-sugar carbohydrate can be categorized. Look at the categorization. We have the polysaccharides and we have the mucopolysaccharides. Now, there's a small tiny difference between the polysaccharides and the mucopolysaccharides. Now, what's the difference? The polysaccharides are made of repeating units of monosaccharides from 11 and above. Now, large amount when compared to the oligosaccharide. No, oligosaccharide is just 3 to 11. Now, talking about the polysaccharide, repeating units of monosaccharides. Mucopolysaccharides are made of repeating units of disaccharides. Be very careful. So, look at it. Repeating units of disaccharides from mucopolysaccharides. Repeating units of monosaccharides from polysaccharides. Now, in this breakdown, that's why in this class we're going to be breaking it down so you understand it properly. Now, these polysaccharides are actually made of 2. We have the homopolysaccharides and we have the what? The heteropolysaccharides. Now, what are the homopolysaccharides and of course the heteropolysaccharides? Now, the homopolysaccharides are made of same units of monosaccharides. Example, cellulose. Of course, I didn't mention just now. Of course, you can still remember. That. We mentioned of inulin, repeating unit of fructose. We have galactans, repeating unit of what? Of galactose. We have starch, glucose actually. Then we have what? Glycogen storage form. Now, what about the heteropolysaccharides? We are talking about the heparin. Then we have the hyaluronic acid. Now, this hyaluronic acid is, of course, is found in connective tissues, in joints, and all that. Now, this is a quick breakdown of carbohydrates. Now, we have other lecture series that we're going to take that I'm going to come up. So that's why I said click the subscribe button so any video drop you get to get it fire department. There's no time wasting. Now look at it now. These mucopolysaccharides, they are quite different. So quickly, we started with sugar, non-sugar. We look at monosaccharide, look at the disaccharide, look at the oligosaccharides and the units that forms them. Now, all other lecture series coming, we have to look at lipids, how to name fatty acid. So that's why you have to look at it very well. I hope that this video was of help to you. Thank you for watching. Now, do well to follow other of our videos and do well, of course, to always subscribe. Thank you very, very much.